So we'll pick up from where we left off yesterday with regard to the replication. The DNA replication, the whole point of that was to make cells like you guys had mentioned earlier. Okay, so now we're going into the making of cells. However, we really want to look at what they call the cell cycle, which is the whole cell's life. They either call it the cell cycle or the cell's life. There's a small chunk of it called mitosis. The whole purpose of mitosis is to make, it's like basically for cell reproduction, to make new cells. And I always tell you this, I mean, and you can joke about it all you want, but it's like, how much of your life is occupied by reproduction? Not much, truly. And most of your life is getting dressed, eating, drinking, food, clothes, work, school, all that other stuff, and a small chunk of your overall life will be occupied by the actual reproduction. The only problem is when we start talking about mitosis, because it's either new or there's so many parts to it, people usually get all caught up in it going, oh, that's the cell's life, it's the cell cycle, and all this other stuff. So we will touch on that mitosis stuff, but it's not the whole entirety of the cell's life. Most of what a cell's life is what they call interphase, and it's when it's doing its job, when it's just doing its cell stuff. Okay. So we'll start at the top right here. We'll do mitosis is what we're going to deal with today. And we'll do this a little bit differently. I know we did it on the board yesterday. We'll do it like this. Mitosis is cell division, and this is part of our learning target today. Is really about the cell cycle, but mitosis in particular. Mitosis is cell division, and it's when one cell divides into two new cells. Okay, so there's a couple big questions. It's kind of like why divide, and you may or may not have ever thought of that. But like one of those things, like why is a cell so small? Why are they so small? Why can't they get bigger? They can only get to a certain size. There's three big reasons why these cells divide. Okay, so we'll put down why divide. First reason is has to do with size. And the best way to think of it is it's almost like if you guys did stuff in geometry and different mathematics classes when you talk about area and volume. I always think about like elk and deer. If you look at an elk and a deer and you compare the two, a deer is probably about a third of the size of an elk. However, mass-wise, the elk probably weighs 2 to 3 times more than a deer. And it has to do with that surface to volume ratio. Even though the outside isn't nearly that much bigger, the volume increases exponentially. Same argument with like a warehouse versus a house. If you have a house with one door and you only got a few people living there, that's plenty. If you have a giant warehouse with 500 people working there, one door is not going to do it. You have to have more than one port. So that same logic goes in place with the size of the cell. The cell can only get so big because the outside can only get so much stuff in for the cell to function. So once it gets too big, it can't get enough stuff in for it to function. Okay? So when cell gets too big, it can't get enough nutrients, etc. to function. Okay, the next one is one that you're more familiar with. It has to do with repair. So cells will divide for repair reasons. If you have like a muscle damage, bone breaks, or other injuries. Okay, the last one is called uncontrolled growth. Bonus, what's another word for uncontrolled cell growth? It's a common term you hear every day. Starts with a C. Rhymes with Banser. There you go. Rhymes with cancer, okay. Just try not to give it away. So I'll do this. We'll put it in parentheses a little bit. Cancer. It's a term that everybody goes, oh my gosh, cancer. We kind of talked about it yesterday with regard to these little warts or moles that you might have on your body. They're basically cancers, but there's two types. So here, let's do this. Uncontrolled growth or cancer is when cells divide out of control. So let's do this. We'll do two things. You have two types of cancer. You can have benign cancers, which usually refer to like a tumor, and the tumor doesn't usually spread. Yep. Right there. It's a tumor that usually doesn't spread. Then you have malignant growths. And the easiest way to remember this, and I use this too, what does mal mean in Spanish?
Bad.
Bad. Okay, that's where that root comes from. It means bad. This is the bad one. And this is because it can spread and what they call metastasize. I'll spell it kind of big. Metastasize. Brendan?
What is metastasize?
That's what I was going to get to. So watch. Every cell has a lifespan. There are certain ones like your brain cell, your heart cell, they're with you. There are certain heart cells that are with you for your whole life. So when you die, they die also. That is their lifespan. Other cells last a relatively short period of time. The cells in your mouth, like when you bite it down in your cheek and you got a little break there, that might repair itself in 3-4 days, right? I mean, you get back and it's done. You get a little scrape in your leg, maybe it's a little bit longer. It just depends. Most of it has to do with partly with blood flow. Okay, so if it's good blood flow, it can repair itself faster and all that other stuff. Now, every time a cell is done with its life, that lifespan, it will get sent off a chemical signal to the cells around it that say, hey, I'm done, I'm out of here. Can you go ahead and make another copies to replace me? So what cancer cells do is they send off that signal uncontrolled. They're sending off that signal, and all they're doing is sending off signals. And then what they do is they also reproduce. They make a copy of themselves. That other cell also sends off the signal. And then before you know it, you've got a bunch of cells that are all sending off signals to make copies. And so when we take that whole thing, we said mitosis, like reproduction, is this part of your life, a really small part of your life. For those cancer cells, that is their entire life. That is the entire sum of their life. And all they're doing is that. So if it happens in your stomach, around little stomach cells, and you start little cancers, all the energy that is going towards your stomach cells is being diverted to those cancer cells. And what they kind of do in a weird way is they starve out energy-wise the cells that are supposed to be working from doing what they're supposed to be doing. And so that's why it becomes deadly. And what metastasize means is this. Like these little, I've got a big tag right here. As long as it stays intact, it's not a big deal. And you guys may have like little warts, little moles that you have that just stay intact. What's bad is if the cells, because all cells have contact with blood. I'll show you a video, probably try and show tomorrow, where the cells, cancerous ones that are sending off that uncontrolled growth signal, they'll break off and go in the bloodstream. And then they'll just land somewhere. They may land in your heart, they might land in your kidney, your pancreas, whatever. They'll land somewhere else, and then they start growing again. But because they're prone to breaking off, they'll tend to keep breaking off. And chunks from the next tumor will break off, and chunks from the next tumor will break off. And metastasize means it's in the bloodstream, it's going all around your body. And when it does that, you can't, if it's a tumor, I'll cut it out. If it's metastasized, it means it's flowing through your bloodstream and it's going throughout your body. You can't nail it down. That's when they have to go to chemotherapy or radiation therapy, usually chemotherapy to try and kill those cells. And again, when it plants the seeds in one of those spots, it starts to starve out energy-wise the healthy cells. And it's hard to target all those things, and eventually you succumb to the good cells not being able to get enough energy and being able to carry out their normal daily functions. That's why if you just see somebody who has cancer, even before they have chemotherapy, one of the common symptoms beforehand is they're just like really tired, like you've had the flu for a while, that type of thing, because maybe because some of the organs or whatever it's involved with, where the cancer is, could be causing a problem with your body functioning and you not being able to process energy as well, and because the cancer is starving out the healthy cells. Ray?
Could you starve out the cancer cells?
That's the whole idea. The whole idea behind chemotherapy is this. It's a chemical that it's usually supposed to be pretty specific to cancer cells, but the idea is this. There's way more healthy cells than cancer cells. I'm going to give you this chemical that should kill the cancer cells, but what they also do in turn is they usually kill healthy cells as well. So what you're trying to do is, hopefully I kill more cancer cells than healthy cells in the process, and I don't kill you before I kill all the cancer cells. I know it sounds bad, but in reality that's really what it is. And that's why when you hear somebody, does anybody know anybody who's had chemotherapy? When they go for multiple rounds, they go in for one, they check to see if you have the cancer cells are still there, and then they wait till you get better, because it takes you a little while. Like, I just killed a bunch of your cells, like maybe your stomach cells. That's why you puke and stuff and your hair falls out, because you're starting to kill cells that are healthy and supposed to be doing normal cell functions. When you get better, they usually wait 2 or 3 weeks till you're feeling better, they give you another round. Let's do it again. And they just keep bombarding you with a chemical. Yes, I mean, there's nothing to do. It is what it is. That's why, if you just have a tumor, it's not that bad. When it metastasizes, starts spreading, it's very difficult. Okay? So that's what all that is. You can have the 3 reasons. We have either size, we have normal repair, or you can have that uncontrolled cell growth. Okay? All right, let's keep going. I'm just going to make a little break here and kind of go. I'll jump back to mitosis in a minute, but I want to talk about the cell cycle, or what they call the cell life. Oops, I put cycle. There we go. Let me cross it out. Cycle. Cell cycle or the cell's life. And essentially there's like, there's 3 big phases. There's 3 big phases. And I'll put phases in like quotation marks here. And I'm going to draw a circle. Hopefully it's not too wobbly. And I'm going to cut a sliver out of this pie. It's a really thin sliver, okay? Just like literally a line. Okay, here's your 3 cycles, or 3 phases. You either have interphase, mitosis, and the last part of the cycle is called cytokinesis. Cytokinesis. I'm going to take and I'm going to make a loop starting from this side of the line all the way around to just the beginning of this side of the line. Right? Then I'm going to draw an arrow from the 1 to here, because that is the time of interphase. Inter means kind of like means between. This is the phase between reproductive events or division events. This interphase is about 98% of a cell's life is spent in just an interphase. This is when you have the cell growing and just doing its job. Whatever its job is. If it's a skin cell, it's doing a skin cell job. If it's heart cell, it's doing a heart cell job. But 98, when it's doing cell division, it's not doing anything except dividing. Once it's done with that, we go back into our cell life and we do our cell thing. Okay? Which is most of its life, 98% of life. Okay? Mitosis is just a small 2% of the cell's life. Right? And I'm going to draw a line all the way around. That is where this little sliver comes out. Cytokinesis is when so here's the thing. Mitosis, you have to do a lot of, you're trying to make the cells separate. When the cell actually separates, like the membranes separate, That's called cytokinesis. And that's at the very end. I'm going to make one more loop that goes up, and it's at the very end of mitosis. I'll show you one more diagram tomorrow, but I think you get the idea. The other thing is everybody gets caught up in the mitosis again, not paying attention to cell cycle. Then they get caught up with the mitosis again, and they go, hey, I just drop back and go, hey, what is DNA replication? They're like, what's DNA replication? Make copy of new DNA. For what purpose? To make new cells. So before we can go through mitosis to make a new cell, we have to make the DNA, right? Okay, so watch. Somehow put it in here. I'm right in this spot just before interphase. I want you to write down DNA replication. We're going to put it at that spot just before we go into interphase. I'm going to flip this sheet.
No.
Are we ready?
No.
No.
Okay, ready? Okay. There's an easy way to remember all these stages, okay? Cell cycle. Flip it over. And I learned this one in college, and I just remembered it's really good, and somebody taught somebody else. It's called pro, mat, cyto. Now, you don't know all of this yet. Some of you guys remember, some of you guys might be able to recall. I stands for what? Interphase. So I'm going to write that down the edge. Pro, you may remember the name of it? Prophase, good. You got prophase. M?
Metaphase.
Good. Metaphase. A?
Metaphase.
Anaphase. Sharpen your pencil, you're good. T?
Telophase.
You can call it telophase or telophase. I usually call it telophase. And the last one is your cytokinesis, okay? So it's really not that hard to remember it. You just got to remember, this is of the entire cell's life, and this part right here, this is of mitosis. This is that second stage, if you will. So I always say, what are the stages of mitosis? And they're like, pro-mat. What are the stages of cell's life? I pro-mat cyto. Just got to remember that. Pretty easy way to remember it. Okay, so look, we're going to prepare for mitosis. We're not in there yet. We're right at the end of interphase, going into mitosis, preparing for mitosis. Yeah. Let's do this. Prepare for mitosis. We got to do this. We got to duplicate DNA. So we're going to duplicate DNA. Once that happens, your chromatin is condensing. And what it does is it kind of goes like this. Once the DNA condenses enough, you can't see DNA in the nucleus. But once it's duplicated and it begins to condense, it starts to look like the spaghetti. Okay, the chromatin starts to look like a spaghetti. Condensing. Chromatin's condensing and it starts to look like spaghetti a little bit. And here's going to be the progression. It's going to go from chromatin, so I'm going to draw a little arrow right here. This is your chromatin. Chromatin's going to condense more. It's going to kind of look like a little squiggly line like this. And it'll have this thing in the middle called a centromere. We're not done yet though. Because we made 2 copies of the DNA that are all together. Remember, I'll let you guys ready. Remember we said our normal chromosome looks like this?
Yeah.
Okay. Because you have 2 copies of the DNA, you actually have 2 chromosomes. They're going to come together and look like a double chromosome. It looks like an X shape. I don't know if you guys have probably seen that before. Most people think that that's what a chromosome looks like. That's when you have a double chromosome. So eventually it's going to get to something that looks like this. Okay? And this is called a double chromosome. Okay? The 2 chromosomes, because they are identical, double chromosome, I'm going to write this. Both sides are called sister chromatids because they're both basically the same thing. Sister chromatids. So eventually you're going to have to separate those sister chromatids to get one on one cell and one on the other cell. Okay, here's what we got. We'll do the stages of mitosis. I'm going to draw. We're going to go to prophase in our first phase. I'm going to draw a cell. What? Good. First thing that's going to happen is we've already talked about the chromosomes thickening. I can't use 23 pairs of chromosomes, so I'm just going to make 2 pairs. I'll do, and they kind of just all kind of squiggly at this point. Okay. 2. There's 2 pairs, and here's 2 pairs. One from mom, one from dad, both sides. But they're also going to converge. They're going to condense all the way up to the double chromosome. I also have the our friends the centrioles finally are going to be here. So centrioles move to poles. So kind of like North Pole, South Pole. We're going to move to the end. So I'm going to put a centriole here. They're at a 90-degree angle to each other. They look kind of like a T almost. So you have centrioles move to poles. They double up at this point too. This is probably the most crucial part. Nuclear. And this stage has the most stuff happening, by the way. Nuclear membrane disappears. So what I'm going to do is I'm going to draw like a dotted line around it because our nuclear membrane was that second line of defense to help protect the DNA. So now that we're getting rid of it, which we have to do so we can separate the chromosomes, we now just lost a layer. Now your $1 million is sitting on the kitchen table. You know what I mean? Doesn't mean anything's going to happen, but it's more likely to happen at this point. Last thing, spindle fibers appear. So they're like little spiderwebby things. These are what's going to actually grab onto the middle part of the chromosomes and pull them apart. Ready? Yes, no?
Yeah.
Okay.
Yeah.
10 more seconds. The last three will go pretty quick. We'll be able to finish. Okay, I'm going to move it up. I have it for later if you really, really need it, okay? Okay, so we're going to go from prophase. Pro means first. Metaphase. Meta means middle. Means middle phase. Because it means like it's going to be lining up in the middle. So I'm going to draw my cell. It says double chromosomes. Line up in middle. So I'm going to do my 2 chromosomes. 2 pairs, I should say. And they don't necessarily stack up evenly, they just stack up randomly, but they all do go down the middle. I'm going to take a little dot and put it in the middle. That's my centromere. There's a little thing in the middle called a centromere. That's what the spindle fibers are going to hang on to. So spindle fibers attach. So what else do I need to draw on here? Spindle fibers, which are attaching to the centrioles, but what are they attached to? What are these things? Centrioles. Okay. So I'm going to draw a line to each middle. It's okay. Just checking. Now here's the other one crucial time. If you have other things like UV rays and such that can knock out nucleotides, the UV rays, here I'm going to put an example. Example, UV can break spindle fibers. So if I break one of those sides of the spindle fiber, if those double chromosomes are supposed to split up, what happens to the number of chromosomes that go on each side of the cell? Yeah, now you have an unevenness. So now you have some issues that can creep up. So this is why mitosis is probably the most vulnerable time of a cell's life, because you got a lot of issues that need to be sorted out and adjusted. All right. Next phase is what?